Hi, I'm Rose Glass. I'm a fifth-year neuroscience PhD candidate at UNC Chapel Hill. I'm part of Jason Stein's lab, which studies early human brain development and how that might change to increase risk for neuropsychiatric disorders. And today I'll be showing you a little bit about one of our projects that uses induced pluripotent stem cells to model the early human brain, and I'll show you all of the equipment that I use in order to actually perform these studies. Development starts from pluripotent stem cells, which will go on to produce all the cells in the body, including the brain. In development, the brain looks like a complicated tube-like structure that's filled with progenitor cells. Those progenitor cells produce neurons that then form the cortex, and the cortex continues to mature up until young adulthood. But in particular, my project focuses on a cohort of these really cool kids who've had their brains imaged throughout infancy, so we know what it grew like immediately after birth. These same kids have now donated a little bit of blood so that we have their cells. These cells are taken into a lab where they're molecularly reprogrammed into what's called an induced pluripotent stem cell. These induced pluripotent stem cells can produce a lot of different cell types of the body, including brain cells. Brain cells are what I grow in Stein lab. I grow them as organoids, which are these 3D cultures that are self-organized. This is what induced pluripotent stem cells look like. They're adherent to a plastic dish and are flat, but we need them to be 3D to make an organoid, which you can see in this well. And then you've got a little bit of kind of faint looking debris, which we get rid of by transferring them into a gel that also helps support their growth into little tube-like structures. Day 36, up through the end of their differentiation, they're in a miniaturized spinning bioreactor, and they grow really well in the bioreactor. To make sure that they actually are doing what we think they're doing, we stain for different proteins that label different kinds of cells. This is a video showing that staining. The light cyan color marks progenitors, and those green circles mark lumens showing their organization that you can also see at day 84 as those bright white circles. This is where cells are grown. It's called tissue culture, and it has a hand washing sink. And then we have something called a bead bath, which has all these beautiful shiny beads that warm up anything that would touch the cells, such as their media. This is a vortexer on the right and a microcentrifuge on the left. Microcentrifuge is used to spin down tubes, which you can see here. This is my lab notebook. It's electronic, and it's where I keep track of all my experiments. These are just big calendars for when I need to do certain steps. And lastly, we have an inverted microscope that has eyepieces and a camera to look at the cells. This is a refrigerator for storing reagents long term. It also has a freezer. This is another centrifuge used for spinning down tubes that are larger. It can also spin them at a bunch of different speeds. This is a biological safety cabinet. It's pretty big, and it's sort of a special box that keeps our cells separate from anything else that might want to eat their media. In here I have a pipette tip set, as well as another smaller microscope for looking at the cells inside the tissue culture space. And then I have pipette tips and other reagents I might need. And lastly, I have some specialized waste containers for biohazardous waste. Now I'm going to show you how to build a miniaturized spinning bioreactor that the organoids will live in. First, you start off with the motor that has this plastic bridge that's going to be used to connect it to the top of the bioreactor. It's got a little metal output that will connect it to the lid, and a series of cords that will convert the AC current coming out of the wall into a direct current. You can change the direct current that it makes by flipping these sets of wires over at this plug. So I'm going to go ahead and put that back on and make sure that my motor and cord and bridge are all good and ready to go. So now that we've shown you the motor, I'm going to show you how the plastic attaches to the lid, which is through those shafts. a set of nuts, and then lastly a set of screws. These all are going to be used to connect the motor to the lid that then sits on top of this tissue culture plate. It has 12 wells. These are what the organoids actually sit in. And it's very important that they stay clean, so everything here has been sterilized. I'm labeling the bottom of the wells real fast just so I can keep track of my samples more easily. Now that that's done, I'm going to show you the lid of the bioreactor. That striped tape, it's what tells me that this is sterile and good to use. So I'll unpack my lid inside of the tissue culture hood. And on the top of the lid is a series of intersecting gears, and the bottom has a bunch of paddles that actually keep the media turbulent and the 3D cultures in suspension. I'm gonna flip this over to show you where the motor will connect. All the gears have a little hole that the metal insert will fit into to actually turn the gears. And you can see the gears turn the paddles on the bottom of the bioreactor lid. I'm gonna replace the top lid of the dish with the bioreactor lid and then discard the original lid and insert my motor into the gear that's across the bridge. Make sure it's on tight by using a small screwdriver. And before I use this bioreactor, I'll also check to make sure that it's well connected by turning the motor to make sure it turns the gears. So we should be all good to go. Now I'm just using the different components I mentioned earlier to make sure the motor is securely attached to the lid so that it's easy to move around and ready for long-term use. This bioreactor will be good for about a month inside of the incubator, which I'll show you later. Here's a bioreactor that has organoids growing inside of it. In order to more easily look at them, I'll swap the bioreactor lid for a clear, regular tissue culture lid. This lets me pick it up, handle the plate a little more easily, just take a look at the organoids by eye. It's also easier to take this lid off and place it inside the tissue culture hood and still keep everything sterile, rather than having the paddles touch any surface that isn't inside the well. Now I'm going to show you how I look at the cells on the microscope inside the tissue culture hood. You can see the wells, and there they are all magnified on my microscope. These are tissue culture incubators. They're essentially nice metal boxes that keep it at 37 degrees Celsius and usually around 5 degrees CO2. Right now it's at 2.8 because I've been opening it a lot. I've just sprayed my hands with 70% ethanol to keep everything nice and clean before I go into the incubator. And I've now opened a water-jacketed outer door that's pretty heavy just to help the incubator maintain its temperature. Now I'm going to open the secondary door, which is made of glass for easy viewing. There's a couple of things going on in here. First, I have a plate of induced pluripotent stem cells. The stem cells are adhered to the bottom of those wells, so you can't see them, and they're just covered in media. That's that nice pink color. Secondly, I've got a couple of different bioreactors on different shelves. They're also labeled with a blue tape so you can tell them apart. And they're connected up through their motor cords, through a couple of different wires and out of the incubator. There they are in action, spinning some organoids. Look at them go. And lastly, we've got a different organoid protocol that uses a shaker. You can see in the media some little bits moving around. Those are also organoids that are just being shaken in a different way. You can maybe see that little bit of white in the pink media. Those are also organoids being shaken. And with that, we'll go ahead and close out this here incubator and also this pre-recorded lab tour. I'm so excited to meet you guys in person, and thank you for watching.